Dear students, today we are going to discuss on the topic metabolism of nucleotides. This topic includes both biosynthesis and degradation or catabolism of purine and pyrimidine nucleotides. Nucleotides consist of a nitrogenous base, a pentose and a phosphate. The pentose sugar is D-ribose in ribonucleotides of RNA, while in deoxyribonucleotides of DNA, the sugar is 2-deoxy-D-ribose. Nucleotides participate in almost all the biochemical processes either directly or indirectly. They are the structural components of nucleic acids, coenzymes, and are involved in the regulation of several metabolic reactions. Now we are going to discuss on the topic biosynthesis of purine ribonucleotides. Many compounds contribute to the purine ring of the nucleotides. N1 of purine is derived from amino group of aspartate. C2 and C8 arise from formate of N10 formyl THF. N3 and N9 are obtained from amide group of glutamine. C4, C5 and N7 are contributed by glycine. C6 directly comes from carbon dioxide. It should be remembered that purine bases are not synthesized as such, but they are formed as ribonucleotides. The purines are built upon a pre-existing ribose 5-phosphate. Liver is a major site for purine nucleotide synthesis. Erythrocytes, polymorphonuclear leukocytes, and brain cannot produce purines. Let me explain the steps involved in the biosynthesis of purine nucleotides. It is otherwise called as de novo synthesis. First, ribose 5-phosphate produced in the HMP shunt of carbohydrate metabolism is a starting material for purine nucleotide synthesis. It reacts with ATP to form phosphoribosyl pyrophosphate, shortly called as PRPP. Glutamine transfers its amide nitrogen to PRPP to replace pyrophosphate and produce 5-phosphoribosylamine. The enzyme PRPP glutamyl amidotransferase is controlled by feedback inhibition of nucleotides. This reaction is the committed step in purine nucleotide biosynthesis. Phosphoribosylamine reacts with glycine in the presence of ATP to form glycinamide ribosyl 5-phosphate or glycinamide ribotide. N10 formyl tetrahydrofolate then donates the formyl group, and the product formed is formyl glycinamide ribosyl 5-phosphate amino group to produce formyl glycinamidine ribosyl 5-phosphate. The imidazole ring of the purine is closed in an ATP dependent reaction to yield 5-amino imidazole ribosyl 5-phosphate. Incorporation of carbon dioxide occurs to yield amino imidazole carboxylate ribosyl 5-phosphate. This reaction does not require the vitamin biotin and ATP, which is the case with most of the carboxylation reactions. Aspartate condenses with the product in reaction 7, that is in the carboxylation reaction, to form amino imidazole 4-succinyl carboxamide ribosyl 5-phosphate. Adenosuccinate lyase cleaves off fumarate and only the amino group of aspartate is retained to yield amino imidazole 4-carboxamide ribosyl 5-phosphate. N10 formyl tetrahydrofolate then donates a 1-carbon moiety to produce formamino imidazole 4-carboxamide ribosyl 5-phosphate. With this reaction, all the carbon and nitrogen atoms of purine ring are contributed by their respective sources. The final reaction of purine biosynthesis is catalyzed by cyclohydrolase, leads to ring closure with an elimination of motor molecule. The product obtained is inosine monophosphate called IMP, the parent purine nucleotide from which other purine nucleotides can be synthesized. Next we are going to see how AMP and GMP are going to be synthesized from IMP. IMP acts as a precursor for the formation of AMP and GMP. Aspartate condenses with IMP in the presence of GTP to produce adenyl succinate, which upon cleavage forms AMP. For the synthesis of GMP, IMP undergoes NAD+ dependent dehydrogenation to form xanthosine monophosphate. Glutamine then transfers amide nitrogen to XMP to produce GMP. 6-mercaptopurine is an inhibitor of synthesis of AMP and GMP. It acts on the enzyme adenyl succinase and IMP dehydrogenase. The formed nucleoside monophosphates, that is AMP and GMP, have to be converted to the corresponding di- and triphosphates to participate in most of the metabolic reactions. This is achieved by the transfer of phosphate group from ATP catalyzed by nucleoside monophosphate kinases and nucleoside diphosphate kinases. Let me explain the inhibitors of purine synthesis. Folic acid in the form of tetrahydrofolate is essential for the synthesis of purine nucleotides. Sulfonamides are the structural analogues of para-aminobenzoic acid. These sulfa drugs can be used to inhibit the synthesis of folic acid by microorganisms. This indirectly reduces the synthesis of purines and therefore the nucleic acids, that is DNA and RNA. Sulfonamides have no influence on humans since folic acid is not synthesized and is supplied through the diet. The structural analogues of folic acid, example methotrexate, are widely used to control cancer. They inhibit the synthesis of purine nucleotides and thus nucleic acids. Both these reactions are concerned with the transfer of 1-carbon moiety, that is formyl group. These inhibitors also affect the proliferation of normally growing cells. This causes many side effects including anemia, baldness, scaly skin, etc. Till now, we have seen the de novo synthesis of purine nucleotides. Now we are going to discuss about the salvage pathway for purines. The free purines, adenine, guanine and hypoxanthine, are formed in the normal turnover of nucleic acids and also obtained from dietary sources. The purines can be directly converted to the corresponding nucleotides, and this process is known as salvage pathway. Adenine phosphoribosyl transferase catalyzes the formation of AMP from adenine. Hypoxanthine guanine phosphoribosyl transferase converts guanine and hypoxanthine respectively to form GMP and IMP. Phosphoribosyl pyrophosphate is the donor of ribose 5-phosphate in the salvage pathway. The salvage pathway is particularly important in certain tissues such as erythrocytes and brain, where de novo synthesis of purine nucleotides is not operative. A defect in the enzyme HGPRT, hypoxanthine guanine phosphoribosyl transferase, causes Lesch-Nyhan syndrome. Coming to the regulation of purine nucleotide biosynthesis, it is well coordinated to meet the cellular demands. The intracellular concentration of PRPP regulates purine synthesis to a large extent. Thus, in turn, is dependent on the availability of ribose 5-phosphate and the enzyme PRPP synthetase. PRPP glutamyl amidotransferase is controlled by a feedback mechanism by purine nucleotides. That is, if AMP and GMP are available in adequate amounts to meet the cellular requirements, their synthesis is turned off at the amidotransferase reaction. Another important stage of regulation is in the conversion of IMP to AMP and GMP. AMP inhibits adenyl succinate synthetase, while GMP inhibits IMP dehydrogenase. Thus, AMP and GMP control their respective synthesis from IMP by a feedback mechanism. Ribonucleotides should be converted to deoxyribonucleotides in DNA. The synthesis of purine and pyrimidine deoxyribonucleotides occurs from ribonucleotides by a reduction at the C2 of ribose moiety. This reaction is catalyzed by a multi-subunit enzyme called ribonucleotide reductase. The enzyme ribonucleotide reductase itself provides the hydrogen atoms needed for reduction from its sulfhydryl groups. The reducing equivalents in turn are supplied by thioredoxin, a monomeric protein with 2 cysteine residues. NADPH dependent thioredoxin reductase converts the oxidized thioredoxin to reduced form, which can be recycled again and again. Thioredoxin thus serves as a protein cofactor in an enzymatic reaction. How the deoxyribonucleotide synthesis is regulated? Deoxyribonucleotides are mostly required for the synthesis of DNA. The activity of the enzyme ribonucleotide reductase maintains the adequate supply of deoxyribonucleotides. The drug hydroxyurea inhibits ribonucleotide reductase by destroying free radicals required by this enzyme. Hydroxyurea is used in the treatment of cancers such as chronic myelogenous leukemia. The next topic is degradation of purine nucleotides. The end product of purine catabolism is uric acid. The nucleotide monophosphates AMP, IMP and GMP are converted to their respective nucleoside forms, that is adenosine, inosine and guanosine, by the action of nucleotidase. The amino group either from AMP or adenosine can be removed to produce IMP or inosine respectively. Inosine and guanosine are converted to hypoxanthine and guanine by purine nucleoside phosphorylase. Adenosine is not degraded by this enzyme, hence it has to be converted to inosine. Guanine undergoes deamination by guanase to form xanthine. Xanthine oxidase is an important enzyme that converts hypoxanthine to xanthine and xanthine to uric acid. This enzyme contains FAD, molybdenum and iron and is exclusively found in liver and small intestine. Xanthine oxidase liberates hydrogen peroxide, which is harmful to the tissues. Catalase enzyme then cleaves hydrogen peroxide to water and oxygen. Uric acid is the final excretory product of purine metabolism in humans. Uric acid can serve as an important antioxidant by getting itself converted to allantoin. It is believed that the antioxidant role of ascorbic acid in primates is replaced by uric acid, since these animals have lost the ability to synthesize uric acid. Most animals, however, oxidize uric acid by the enzyme uricase to allantoin, where the purine ring is cleaved. Allantoin is then converted to allantoic acid and excreted in some fishes. Further degradation of allantoic acid may occur to produce urea and later to ammonia. Students, let me explain the disorders of purine metabolism. Uric acid is the end product of purine metabolism in humans. Whenever the concentration of uric acid in blood increases, that condition is clinically called as hyperuricemia. The normal concentration of uric acid in the serum of adults is in the range of 3 to 7 milligram per deciliter. In women, it is slightly lower than in men. The daily excretion of uric acid is about 500 to 700 milligrams. Hyperuricemia refers to an elevation in the serum uric acid concentration, as I have said earlier. This is sometimes associated with increased uric acid excretion, clinically called as uricosuriia. Gout is a metabolic disease associated with overproduction of uric acid. At the physiological pH, uric acid is found in a more soluble form as sodium urate. In severe hyperuricemia, crystals of sodium urate get deposited in the soft tissues, particularly in the joints. Such deposits are commonly known as tophi. This causes inflammation in the joints, resulting in a painful gouty arthritis. Sodium urate or uric acid may also precipitate in kidneys and ureters, that results in renal damage and stone formation. Historically, gout was found to be often associated with high living, overeating and alcohol consumption. In the previous centuries, alcohol was contaminated with lead during its manufacture and storage. Lead poisoning leads to kidney damage and decreased uric acid excretion, causing gout. In general, a diet rich in meat and seafoods is associated with increased risk of gout. The prevalence of gout is about 3 per 1000 persons, mostly affecting males. Postmenopausal women, however, are as susceptible as men for this disease. Gout is of two types, primary and secondary. Primary gout is an inborn error of metabolism due to overproduction of uric acid. This is mostly related to increased synthesis of purine nucleotides. The metabolic defects found in primary gout are: the first enzyme is PRPP synthetase. This enzyme is under feedback control by purine nucleotides. However, variant forms of PRPP synthetase which are not subjected to feedback regulation have been detected. This leads to increased production of purines. Other enzyme is PRPP glutamyl aminotransferase. The lack of feedback control of this enzyme by purine nucleotides also leads to their elevated synthesis. HGPRT deficiency. This is an enzyme of purine salvage pathway and its defect causes Lesch-Nyhan syndrome. This disorder is associated with increased synthesis of purine nucleotides by a twofold mechanism. Firstly, decreased utilization of purines by salvage pathway, resulting in the accumulation and diversion of PRPP for purine nucleotides. Secondly, the defect in salvage pathway leads to decreased levels of IMP and GMP, causing impairment in the tightly controlled feedback of their production. The next enzyme deficiency is glucose 6-phosphatase. In type 1 glycogen storage disease, glucose 6-phosphate cannot be converted to glucose due to the deficiency of enzyme glucose 6-phosphatase. This leads to increased utilization of glucose 6-phosphate by hexose monophosphate shunt, resulting in elevated levels of ribose 5-phosphate and PRPP and ultimately purine overproduction. Von Giertz disease is also associated with increased activity of glycolysis. Due to this, lactic acid accumulates in the body, which interferes with the uric acid excretion through renal tubules. Elevation of glutathione reductase generates more NADP, which is utilized by HMP shunt. This causes increased ribose 5-phosphate and PRPP synthesis. Secondary gout. Secondary hyperuricemia is due to various diseases causing increased synthesis or decreased excretion of uric acid. Increased degradation of nucleic acids is observed in various cancers like leukemia, polycythemia, lymphoma, etc., and in psoriasis and increased tissue breakdown. The disorders associated with impairment in renal function cause accumulation of uric acid, which may lead to gout. By administration of uric acid isotope N15, the miscible uric acid pool can be calculated. It is around 1200 mg in normal subjects. Uric acid pool is tremendously increased to 3000 mg or even more in patients suffering from gout. What is the treatment for gout? The drug of choice for the treatment of primary gout is allopurinol. This is a structural analog of hypoxanthine that competitively inhibits the enzyme xanthine oxidase. Further, allopyrinol is oxidized to alloxanthine by xanthine oxidase. Alloxanthine in turn is a more effective inhibitor of xanthine oxidase. This type of inhibition is referred to as suicide inhibition. Inhibition of xanthine oxidase by allopyrinol leads to the accumulation of hypoxanthine and xanthine. These two compounds are more soluble than uric acid, hence easily excreted. Besides the drug therapy, restriction in dietary intake of purines and alcohol is advised. Consumption of plenty of water will also be useful to the patients. The anti-inflammatory drug colchicine is used for the treatment of gouty arthritis. Other anti-inflammatory drugs such as phenylbutazone, indomethacin, corticosteroids are also useful. Pseudo-gout. What is pseudo-gout? The clinical manifestations of pseudo-gout are similar to gout, but this disorder is caused by a deposition of calcium pyrophosphate crystals in joints. Further, serum uric acid concentration is normal in pseudo-gout. Hypouricemia. Decreased serum uric acid levels is called or represent hyperuricemia. Decreased uric acid levels in the serum, that is less than 2 milligrams per deciliter, represent hypouricemia. A rare genetic defect in the enzyme xanthine oxidase. It leads to the increased excretion of xanthine and hypoxanthine. Xanthinuria frequently. In this class, we have discussed about de novo synthesis and salvage pathway of purine nucleotides and as well as catabolism of purine nucleotides. Under catabolism, we have also discussed a clinical disorder called gout, which has been classified into primary and secondary gout, and what are the reasons behind that and how could it be treated. Everything we have discussed. So in the next class, we'll be discussing about biosynthesis and degradation of pyrimidine nucleotides. Thank you.